First, what will happen? The parent hydra, which will be well matured. So some part of that, you can notice that there is a repeated mitotic divisions will take place. And we can notice a small bird will form. And then this bird will start developing its mouth and as well as the tentacles. You can notice it. So it will turn up into new hydra. But still, it not detached from the parent. So once it is completely developed into a hydra only, it will be started detaching. So it will turn up into complete hydra, complete hydra. All the mouth and as well as tentacles, everything forms. And finally, this new hydra, the small, tiny hydra, it will be separate out, detaches itself from the body of a parent, and it will be, you can say, new hydra will separate out from the parent and leaves it as a separate organism. So that is how the parent hydra has produced a new hydra, and again this new hydra will be grown up. Once it will be turned up into enough size, again the same process will be takes place. So that is how the hydra reproduces asexually by growing birds from its body. So that is why this process we used to call as, what is the process we used to say? We used to call it as budding. Understanding? So that is what you have to remember. But you have to, one thing you make it here, bird is formed in a hydra is not a single cell. The bird is not a single cell. It is a group of cells. It is not a single cell. It is a what, everybody? It is the group of cells because it is a simple multicellular animal. It is not the single cellular animal. That is what you have to remember. So any doubt, anybody can ask me. Are you clear, everybody? All of you? So learn this diagram also. This is how the bird formation, with the help of a bird formation. So the hydra animal, simple multicellular animal, will continuous its offsprings or daughter cells. Okay, so let us watch a small video regarding this and then we'll proceed. The second organism which will undergoes this budding. What is the second organism which undergoes this budding? Everybody?
Yeast.
Yeast.
Okay. So let us see that actually. So yeast is a what actually? Animal or plant? Yeast is a plant or animal. Everybody? Which is a non-green plant, which has been a family of fungus. Fungus family it is. What family it is? Everybody? Fungus family. We'll discuss it. Okay.
Parent organism grows in budding, a small part of the body of the parent.
Yeah. So small part of the body will stop from the parent organism, grows out as a bird, which will then detaches and becomes into a new
organism grows out as a bud, which then detaches and becomes a new organism. Example, in Hydra, first a small outgrowth called bud is formed on the side of its body by the repeated mitotic divisions of its cells. This bud then grows gradually to form a small hydra by developing a mouth and tentacles. And finally, the tiny new hydra detaches itself from the body of parent hydra and lives as a separate organism.
Are you clear, everybody? Understand, Amma? All of you? So that is what, how that hydra will be turned up into the new organism. So now, let us take about the next organism. So I hope you all understanding. So hydra is the simple, multicellular animal. Now coming to the yeast, you can see the reproduction in yeast plant by the process of budding. So this is actually a non-green plant. So basically yeast we'll use in our daily life also. So many purposes we'll use it. You might be notice it. So those who are baking industries and all, so the yeast is actually produced. Yeast powder we will use it. So even yeast culture solution in even laboratory, if you notice it, we use yeast for many purposes in our daily life. So what is this yeast and how it will be produced? Let us discuss now. So if you take here, the single cell of a yeast is actually a complete plant itself. Actually, it is unicellular. It's not a multicellular. A single cell itself is a complete plant actually. So yeast belongs to the plants, but what plants it is? Green, non-green plants. Okay. So yeast is the actually a single cellular, or we can say single celled or unicellular non-green plant. Write it. Non-green plant, which is a what family, guys? Non-green plant, which is a what family? Fungus family. Okay, belongs to fungus. Now, this yeast is also produced by the process called as budding only. So, a single cell itself is called as a complete plant, parent plant, we used to say. So, please remember that. Okay. So, yeast is also produced by the what method? Everybody? By the method of budding. Okay. So single cell itself is called as a complete yeast. Now let us discuss how this will be taking place. Now here also same thing. So a parent yeast cell, which is actually a complete plant. So first a bud appears on the outside of the cell wall. You can notice it here. So this is the complete parent yeast cell. So first what will happen? We can notice a bud which appears outside the cell wall. So bud is forming, you can see. Again by the same thing, the cell divides into two parts and one part of the nucleus moves into the bud and everything. See that the nucleus will be dividing into two parts. Cytoplasm is also spreading. So everything it will be turned up into new bud. So ultimately the bud will separate off from the parent yeast cell and it will form say new yeast cell. You can see the bud niped off. What do you mean by niped off? Anybody? Removed or detached, we can say. So it is detached from the parent yeast cell. So that is what we used to say. New yeast cell will form. Or the buds remains attached, you can see, and they will form long chain of yeast cells will be formed. So that is how it will be taking place. So the bud will separate off from the parent yeast cell and they will form into a new yeast cell, or we can say new yeast plant, because a single cell itself is to called as a plant. So this budding in yeast, you can see it will takes place very, very fast. Understanding all of you? So the first bud starts forming their own buds and all of them remains attached to the parent yeast cell, forming a long chain. You can notice it. So yeast cells. So that is how after sometimes all the yeast cells of the chain, they will separate from one another and they will form into a individual yeast plants. So extremely faster, the fission will take, the budding formation will be very, very faster. So that is why you can see from the parent, one will form, that one again separating into another one, that one will be another separate one, another one into another one. So like that multiple continuously, very fast growth will be taking place. So the first bird starts forming their own birds and all of them remains attached to them. So what forms by the parent yeast cell, this bird will be start forming the another daughter cell, another daughter cell. So they will be continuously flow, it will be takes place. That is how they'll form a long chain of yeast cells. That is what we can say yeast powder. We can notice in our lab also. So that is why after sometimes only they will be started detaching from each other and they'll continue again the process of budding. So continuously nucleus will split, continuously the new daughter cell will form. That daughter cell again it will be turned up into a new daughter cell by formation of buds continuously. So that is how the yeast will be produced. Even our laboratory, we can produce the yeast. Even market it is available. You can see. We will use this yeast in our for food items also. So that is how it will be taking place. The yeast culture even we can take in our laboratory also. We can produce it. So we can study this process of asexual reproduction yeast by budding in laboratory. So what is the required things for this one? So if you need to take 100 ml of water in a conical flask. You all people knows a conical flask, how it will be. So you can take a conical flask. Yeah, baking, they call it as activation of yeast. Yes, Asini. Baking industry, they will use this yeast for many things. Okay. So basically, if you want to see this cultivation of this yeast in the lab, asexual reproduction, just take 100 ml of water in conical flask and dissolve just 10 grams of sugar in it. Clear, everybody? So 100 ml of water and dissolve 10 grams of sugar in it. Then add 5 grams of yeast. So why we are adding sugar here? Anybody can tell me? Why we are adding sugar here? Please respond. What is the purpose of sugar? Food, guys. They need food. Do you understand it? So that is what is the glucose. Sugar will contains glucose. That is the reason. So add 5 grams of yeast powder or yeast granules to the sugar solution and you stir it with a glass rod. Take one glass rod and you just stir it. Then put a cotton plug in the neck of the conical flask. Like a cotton, you take it and you put it at the opening end of the conical flask. Then you can see the conical flask contain the sugar solution. So you prepared sugar solution by dissolving 10 grams of sugar in 100 ml of water, and also you added the yeast. So what will happen? The yeast which is present inside this one, actually there is no outgoing or like air is not exchanging because I kept a cotton plug. So they're completely closed. So you just keep this one in a warm place for 3 to 5 days. 3 to 5 days, you can place it. Even you can market it is available. You buy and you can try. You can see when you open, like when observed in the flask, the yeast culture is ready for the after 3 to 5 days. If you observe, the yeast is already grown and you can observe with the help of microscope. So take a small quantity of the yeast culture solution. The entire solution which has been prepared, we used to call it as yeast culture solution. So take small quantity because we have prepared 100 ml of solution now. So take a small quantity, okay, from the near the bottom of the conical flask with the help of a dropper or place a dropper inside the cultural solution and you can take like with the help of a droppers, we all people know. So take a small amount of from the bottom of the conical flask and add it to the slide. So what is a slide? Microscope slide. So a clean, clear microscope slide, you take it. With the help of dropper, you just put it on it. Now, very small amount of, for this slide, you just add a small amount of iodine. So we all know iodine we have used in biology, first chapter also. Where we have used, guys, iodine solution? Anybody? Iodine
Sir, to
solution
check
we
the
have used it. Yes. To check the what, Harish?
starch, presence of
Yes,
starch.
starch present. So for this slide, so few drops of the solution, yeast culture solution you have to add and add few drops of iodine to it. So what will happen, all of you? So you can see here, once if you add iodine and just place one more slide over this one, means like between that, the solution is there between the two slides. Now you keep this slide under the microscope and observe the with a low microscope, and then slowly increase the high power of microscope. You can see the formation of birds in the yeast cell, how they are separating from the parent cell. The similar picture, even when I'm in 11th class, when I'm studying in my university, so 11th class and 12th class practicals time, I used to form this one. So even one of our PUC course, PUC pre-university courses, when we were studying time, we used to see this one. This is one of the one of the biology lab actually in 11 and 12. So we need to see the proper formation of bird formation in the yeast cells. We can exactly notice the long chains formation and all. So that is how the bird formation will take place. We can very clearly notice the microscopic pictures, how the one cell will forming into a bird, another, the single cell will be another turn up into daughter cell, another daughter cell formation like this one. Exactly we can notice in the under powerful microscope, high power of microscope. So that is how you can see. Even in your home also you can make and you can prepare the slides, and the slides can be viewed through the microscope from our school laboratory. You can all can try also. Even I think in our school, you can do this is as a project also. So that is how the budding process only, the yeast powder, which we are using in, basically we are preparing, the yeast powder is used for making yeast culture solution in the laboratory. So we can notice how the budding formation is started helping to grow the yeast. Okay. So even like this yeast, some other organisms also, you might be heard organisms like sponges. So sponges also undergoes this one, or corals also. Okay. Corals also, they will undergoes, like the birds remains attached to the parent organism permanently. So they also will by growing by the birds, but here the birds are attached permanently to the parent organism. Please note it, this point. So sponges and corals, the birds remains attached to the parent organism very permanently. So these birds then grow and produce birds of their own. Like birds will produce their another birds, but another birds will produce to the another birds. So finally, all will attach to the parent organism only. So that is how you can see the colony of sponges or corals is formed, because the birds will never separate out from the parent organism. That is why we can see a long chains will form. That is what we used to call as colony of sponges, or we can say colony of corals is formed. Because why, guys? The birds not separated out from the birds permanently attached to the what? Yes, please. Permanently attached to the
Parent
attached to parent organism. That is what it will be happening. So this is how everything will be takes place in the budding. I will show you that organisms also. You can see that. So might be not seen. So you all can notice it. The colonies of these sponges. So you can see that bird formation of sponge. You can notice it, how it will be takes place. That is how the images also you can notice. The bird formation of sponges will be same. But the thing is, what will happen here? All the birds will be permanently attached to the parent organism. So this is what the structure of the formation of the sponges and all it will be takes place. So next you can see that another one. So what is that? Another one, corals. So corals also you can see the same thing will be taking place. So all the birds will be attached to the parent organism. So that is how it will be taking place. So that you can notice it. The colony of corals. Okay, so that is how it will be taking place. See that it will be long. Every bird will be attached to the parent itself. Clear, everybody? Understand, Amar? At least have a idea somewhere. If you notice it, you can identify it. What is that? So colonies of sponges, you can see that. So each bird will be attached to the same parent organism. You can notice it. Clear, everybody? All of you? So this is also will formed by the budding method only. But here what will happen? They will be attached to the main parent permanently. That is the only difference it is. So this is how it will be taking place. This is the second asexual reproduction method. Okay, second asexual reproduction method, that is what budding. So under budding, two organisms you should learn. The diagrams permanent, everybody has to learn the diagram. So what is the diagrams, everyone? All of you. One is formation of hydra, and another one is what? Budding in hydra, and the second one is what? Formation of yeast cells. Yeast cells in the, but you can see hydra is the single multicellular, simple multicellular organism. But yeast is a what? Everybody? Yeast is the unicellular non-green plant which belongs to a fungus family. So that is what you have to remember. Then tomorrow we'll study spore formation. Spore formation is one of the most important asexual reproduction method. Even many times the diagram is also asked in our board exam also. So spore formation and then regeneration. So both will try to finish on tomorrow's class, then fragmentation and vegetative propagation. If it is over, then artificial propagation of plants. So artificial propagation of plants is over, half of the chapter will be completed. Mostly by this week, we'll try to finish this up to asexual methods in plants. Then we'll go with the sexual reproductive methods in animals. Okay. So it's already 6:04. Get back to by 6:10, 6 minutes time, and start working. Prepare for your DRD exam or whatever it is. I hope today's school will end early. Am I right? All of you? Correct? Might we receive the message?
Yes, sir.
Yeah.
Sir.
Make use of time. You have a surplus time. So 100%, if you are sincere, you can use it. Avoid everything and try to bring good name. Over August also over. Today, August 23rd. So I'm expecting everybody will show more sincerity. So kindly cooperate it and start working. Don't sleep and don't cheat your parents because they are believing you more and they are making many things for your purpose. Keep on saying, keep on disappointing is not the right way. Okay. Thank you, everybody. We'll meet in tomorrow's class. Please, all of you, work, start to get back to work immediately.